Just imagine, for a second, holding the power to literally rewrite human evolution, to cure diseases we thought were completely incurable, to reshape our environment, maybe even design the biological future of our entire species. You'd probably expect a power of that magnitude to be built in some billion-dollar top-secret underground lab, right? Well, the truth is actually far stranger, because this godlike ability didn't begin with some grand master plan. It started completely by accident, hiding out quietly under a microscope, inside the most humble, ordinary place you could possibly imagine. So picture this. It's 1987. A young Japanese scientist named Yoshizumi Ishino is looking at the genetic code of E. coli. Yeah, just your common gut bug. And as he's scanning the genome, he stumbles across something entirely bizarre. Tucked away in the microbe's DNA are these weird repeating sequences. They're short, they're palindromic, and they're perfectly spaced out by these unique non-repeating segments. And the crazy part? They matched absolutely nothing in any known scientific database. So Ishino publishes a paper pointing out this strange anomaly. And then, crickets. Nobody cared. Literally for over a decade, this colossal secret just gathered dust in the footnotes of obscure microbiology journals, totally ignored by the world. It actually wasn't until 2002 that a Dutch researcher named Ruud Janssen finally blew the dust off this mystery. He's the one who gave these weird sequences a name: clustered, regularly interspaced, short palindromic repeats, or, thankfully, as we know it today, CRISPR. But here's the real secret. CRISPR isn't just some boring, complicated academic acronym. It's actually alive. It is an ancient, microscopic war machine. Here is where it gets absolutely wild. In 2005, 3 totally independent research groups cracked the code, and they revealed this hidden billion-year-old war going on. They realized those unique spacer sequences wedged between the repeating CRISPR clusters, they weren't just random genetic junk. They actually matched the DNA of bacteriophages. Those are deadly viruses that actively hunt and kill bacteria. Suddenly, the impossible just clicked into place. CRISPR is a primitive immune system. Bacteria were literally tearing off pieces of their attacker's DNA and storing them like tiny molecular mugshots in their own genome. Think about that. Single-celled organisms with no brain, no nervous system, were keeping a hit list of their enemies so they could destroy them if they ever dared to return. Fast forward to 2011. French microbiologist Emmanuelle Charpentier is studying a really dangerous pathogen known as Streptococcus pyogenes. And deep inside this lethal bacteria, she finds the missing puzzle pieces to this whole immune system. She discovers this small molecule called TrkRNA, which basically acts like a homing beacon. And working right alongside it is an enzyme called Cas9. You can basically think of Cas9 as a pair of highly programmable molecular scissors. When that homing beacon finds a virus that matches one of those stored mugshots, the Cas9 scissors deploy and they slice the invader's DNA with absolute lethal precision. Okay, so at this point, the story just kicks into overdrive. Later in 2011, Charpentier travels to a conference in Puerto Rico, where she meets Jennifer Doudna, a brilliant structural biologist from UC Berkeley. The two women decide to team up, and their collaboration hits the scientific community like a massive thunderbolt. By June 2012, they publish a landmark paper proving that this bacterial immune system could actually be hijacked. You could engineer it to cut literally any DNA sequence simply by feeding it a new biological mugshot, and the scientific race was on. By January 2013, a scientist named Feng Zhang at the Broad Institute successfully took this exact system and used it to edit human and mouse cells. The genie was officially out of the bottle. To really grasp the sheer scale of this technological leap, we have to look at what came before. Before CRISPR, editing the code of life was, frankly, a total nightmare. Older methods like zinc finger nucleases were incredibly clumsy. They were wildly expensive, and they took entire teams of elite scientists months just to design. CRISPR shattered that paradigm overnight. It's cheap, it's fast, and it works on almost anything. Suddenly, graduate students with basic training could program the code of life in a matter of days for just a few $100. Humanity had just found the ultimate biological search and replace tool. And the applications, they started materializing at a breathtaking speed, pointing straight toward a true medical miracle. Devastating genetic diseases that had stalked human families for millennia, maybe even diseases in your own family, were suddenly sitting squarely in the crosshairs. We're talking hereditary blindness, beta thalassemia, sickle cell disease, which is this brutal, agonizing condition caused by literally a single tiny letter change in the hemoglobin gene, one wrong letter out of billions. And CRISPR now had the power to find it, cut it out, and fix it.
And guys, this isn't science fiction anymore, this is happening right now. In December 2023, the United Kingdom made history. They approved Casgevy, the world's first CRISPR-based therapy. Patients who had suffered their entire lives from excruciating pain crises are being effectively cured. The process is pretty straightforward. Step 1, they extract the patient's bone marrow. Step 2, they edit the cells with CRISPR to reactivate fetal hemoglobin production. And step 3, they infuse those edited cells right back into the body. A single treatment and an entire lifetime of suffering is just rewritten. But right at this incredible high, a massive shadow falls over the whole thing. November 2018. A Chinese scientist named Jiankui shocks the globe with an announcement that literally stops the scientific community dead in its tracks. He had taken those Cas9 scissors and crossed the ultimate ethical line. He used CRISPR to edit viable human embryos, creating the very first gene-edited humans ever born, twin girls named Lulu and Nana. He disabled a gene called CCR5, trying to get the twins' genetic resistance to HIV. 2 babies, carrying altered DNA, permanently introduced into the human gene pool. The outrage was instant, and it was global. Jiankui totally bypassed ethical boards. He used an experimental, unproven technique on human embryos, and he introduced unknown, permanent mutations that these twins will now carry and pass down to their children forever. He was ultimately sentenced to 3 years in prison, but the terrifying reality remains. The line between curing a deadly disease and designing a human being has been breached. I mean, if we can edit out sickle cell, what exactly stops someone from editing in higher intelligence, or greater height, or a specific eye color? And really, who gets to decide? And while we're all wrestling with the massive ethics of human design, this tool isn't waiting around. It's rapidly rewriting the world outside the hospital walls, too. Agricultural scientists are forging drought-resistant crops so we can survive a warming planet. Conservationists are engineering these things called gene drives, designed to actually crash populations of malaria-carrying mosquitoes, which could alter entire ecosystems. Industrial biologists are prototyping microbes that eat plastic waste, and the tool itself keeps evolving. We now have new variants, like base and prime editors, that can literally swap single DNA letters without even severing the double helix. The precision is unimaginable. In October 2020, just 33 years after a lone scientist spotted a weird, repeating pattern in a basic gut bug, Emmanuelle Charpentier and Jennifer Doudna were awarded the Nobel Prize in Chemistry. It stands as one of the absolute fastest journeys from a quirky basic science curiosity to a totally world-altering technology in the history of human civilization. A microscopic defense mechanism completely transformed into a tool for rewriting the code of life itself. We have finally unlocked the source code of our own existence. The pen is in our hands. The only question is, what are we going to write next?